Okay. So my voice is audible. Sorry for the little distraction before. Are you getting my voice? Any one of you, are you getting my voice? Yeah. Hello. Are you getting my voice? So today we have to start our flow cytometry analysis, as well as I will try to start bioinformatics also. So have you heard of flow cytometry before also? Have any one of you have any knowledge regarding the flow cytometry also? Because there are lots of workshops which are there on flow cytometry. So I hope many of you have aware of this term flow cytometry. So today we will focus on flow cytometry, and this is very important technique in MBBT also. So we will keep it very simple. I will let you know the basic concept which is there in the flow cytometry. I will let you know, because flow cytometry is a technique. You have to do it practically only. Then you will be able to understand it more easily, and you will able to do it by practice only. But till we will try our level best, so that all of you will be aware of flow cytometry. I'm just waiting for the students to join, then we will start. So good evening to all of you, and sorry for the in between network issue, but now the signal is okay. It is good now, so I think there will be no buffering right now. So I have to start the session as soon as possible. Okay. So, have you completed your yesterday's assignment or not? Please reply me fast. Then I will start flow cytometry. Have you done your previous assignments also? Because we have these 2 days for completing our theoretical work. After it, you have to focus on your review paper. So I suggest you, I request you to submit your assignments on time. That is the one thing. And today we have to start flow cytometry. I hope you are getting my voice. So I am just starting the presentation, because I don't know when the signal will get off. So I have to start it, so the people is visible to all of you. Everything is fine in the before lecture series. I don't know what happens in this lecture series, why it is buffering right now. So are you able to see this presentation, flow cytometry? Any one of you, please let me know. So this is visible to all of you. So shall we start now? Shall we start now? Flow cytometry will be okay soon. I don't know why this is happening now. Otherwise, in the previous lecture, everything is fine. So shall we start now? And one more thing, today I will try, no, not today, tomorrow I will try to take your class in the morning, because at this time from 9 to 10 p.m. there is so much disturbance in the net. So I will try to take your class in the morning, so that there will be not any buffering and something like that. For Monday, for Sunday, that is for tomorrow, I will try to take your class after, mm, you can say after lunch maybe, but I will try to take your class by 2 p.m. or by 3 p.m. So I am really for the inconvenience for tomorrow, but you have to be there. If you are not, it's okay. I will deliver the lecture and you can watch it later, because at that time there is so much disturbance. I have my class at 3 p.m. Then I have my lecture at 5 p.m. also. There is not any a single time the slides get buffer, but now it's buffering. So that is why I don't want the loss of any of you. So tomorrow I will try to take it in the noon session, because I know tomorrow is Sunday. You all must awake little bit late, so we will try to do it after lunch by 2 p.m. Okay, by 2 p.m. or by 3 p.m. we will try to take it. Okay, and the session will be little longer also, so please be ready with this thing also. So and now I am just going to start the presentation of flow cytometry. I will came back to see whether everything is okay. If it is still buffering or something is like that, then we will try to do it tomorrow also. No worries. But let me just take it once only. If everything will be fine, then we will do it. Otherwise, we will it further. Okay, so now I am just going to start with the flow cytometry. Now this is flow cytometry. A flow cytometry is what basically? Obviously, this definitely will be a technique which is useful in the molecular biology basically. So what is flow cytometry? Let us start it. Hello, Anji. हाँ जी, हाँ जी, गुड इवनिंग. हाँ जी. कौन भूल गए हुए हैं? आप कौन हो? मुझको मैं जानती नहीं होती आपको भी. नहीं 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 नहीं. हाँ. So flow cytometry is the measuring property of a cell as they flow in a fluid suspension. Flow cytometry is, what is the definition of flow cytometry? Flow cytometry is measuring property of a cell as they flow in a fluid suspension across an illuminated light path. So basically in flow cytometry, our main focus is on a cell. Getting my point? Now what is basically a cell? Any one of you have you know what basically is a cell or the organelles which are there in the cell? No worries, we will discuss the timings later. Plus, please now let me know. Have you aware of the thing which is basically this cell, the organelles which are there in the cell? What is basically a cell? Any one of you, please let me know what is cell basically. So what is cell basically? Any one of you please let me know about cell, the organelles which are there in the cell. Please, any one of you please let me know. Is it still buffering or is it okay now? It is still buffering. Now please let me know the cell organelles which are there. What is basically there in the cell? What is cell basically? Any one of you please let me know what is cell. What is cell? What are the organelles which are there in the cell? What are the organelles which are there in the cell? Any one of you? Yes, Vishwa, please let me know what is cell and what is the organelles which are there in the cell. So basically flow cytometry, yeah. So cell is a basic structural, functional, biological unit. And what are the organelles which are present in the cell? What are the organelles which are there in the cell? Yeah, cell is a basic structural and functional unit of our life. So in our main focus is to study the cell basically. The main focus is to study the cell basically. Yeah, microbial news is living particles. Okay. So now I am just starting with flow cytometry. So in flow cytometry, our main focus is basically to study the properties of a cell. In flow cytometry, it is the measuring when we have to measure the properties of a cell as they flow in a fluid suspension across an illuminated light path. So it is the in it flow cytometry we have to study the properties of a cell. So the concept of flow cytometry has been in existence for more than 5 decades. Flow cytometric immunophenotyping first appeared in clinical laboratories in 1980s in the wake of AIDS epidemic. So initially utilized to assess CD4 T cells, the technique was soon applied to lymphoid and eventually to myeloid neoplasm. So in flow cytometry, we are basically working on we just want to understand the cells basically. So this is the basic mechanisms of flow cytometry. We have a biological sample. Let me check everything is fine now. Yes. Thank you all. Thank you, Mahivesh, Vishwa, Shage, Jafrin. Thank you all. I think now the signal is okay. No, it's not okay. Yeah. So basically, in case of flow cytometry, our main focus is on the cells. Basically, we want to understand the cells basically. So, what is the basic mechanism? The biological sample is there. The biological sample is there. We have to label it with a fluorescent marker. Okay, we have to tag it with a marker. After it, the cells, they will move in a linear stream through a focused light. So the cells will move in a linear stream. Fluorescent molecules gets activated and emits light that is filtered and detected by sensitive light detectors. So definitely when it will be passed through the laser beam, what will happen? The fluorescent molecule gets activated and it emits light that is filtered and detected by sensitive light detectors. So conversion of analog fluorescent signals to the digital signal. Then we will convert that fluorescent signal into the digital signal in the computer. So this is basically the basic mechanism which is there in case of flow cytometry. We have to take the biological sample. We have to label this biological sample with some marker. Then this biological sample, the cells, they will move in a linear stream through a focused light, or you can say through a laser beam. These cells will move. And now when they will pass through the laser beam, what will happen? The fluorescent molecules gets activated and they emit the light that is filtered and detected by sensitive light detectors. Then after it conversion, then these analogic signals, these fluorescent signals, they will converted into the digital signals in the computer. So flow cytometry, this method allows the quantitative and qualitative analysis of several properties of a cell. Please keep this thing in mind that in flow cytometry, we are working on the properties of a cell from virtually any type of fresh unfixed tissue or body fluid. So here we are working on the cells only. So the properties measured include a particle's relative size. That means basically we want to know the size of the cell, relative granularity or internal complexity of the cell, and relative fluorescence intensity. So in flow cytometry, our main focus is on three things, three properties of a cell. Number one, we have to know the size of the cell. Number two, we have to know the internal complexity in the cell. Number three, relative fluorescence intensity. So we have to know these three properties of the cell. So mostly analyzed materials are blood. So the cells which are present in the blood, basically those cells are detected. Lymph node suspensions are detected. Getting my point? So what are the three properties on which flow cytometry works? It generally the properties, it generally includes particle size, the size of the cell, number one; internal complexity of the cell, number two; relative fluorescence intensity of that particular cell. So we are mainly focused. So flow cytometry, as the name suggests, is a technique for cell counting and measuring of different properties of the cell. Flow cytometry. Cyto means cell. Metry means count or measure. So in flow cytometry, flow means the cells will moving continuously, and at that time we have to count the cell as well as we have to measure the properties of the cell. So it is a laser based technology that measures and analyzes the different physical chemical properties of the cell flowing in a stream of fluid through the beam of light. So the cells, they are passing through the beam of light. At that time we have to count the cells, we have to measure the cell, we have to understand the different physical and chemical properties of that particular cell. Are you getting me? Are you getting me? Everything is fine now? Hello, are you getting me? So are you getting me? Any one of you, please reply. Okay. Okay, thank you, thank you. So flow cytometry, as the name suggests, is a technique for cell count. First, cyto means cell, metry means counting or measurement. Measurement of different properties of the cell in the presence of laser beam, basically, or light, basically. So it's a computer based technology that measures and analyzes the different physical and chemical properties of the cell particles flowing in a stream of fluid through a beam of light. This is the definition of flow cytometry, that is, in the presence of beam of light, in the presence of light, we have to count the cell, the size of the cell, the internal complexity of the cell, or you can say different properties of the cell. Now this is the machine which is there for flow cytometry. If you are able to see, this is the place where you have to put your sample, and then you have to start the apparatus. You will get all results in this computer only. Okay. So you will get graphs only in case of flow cytometry. From that graphs you have to, let me show you one more diagram. So from that graphs, basically you have to understand the results. So this is the flow cytometry apparatus. In it we have the machine in which we have to put the sample, and all the results, everything will come here in the computer in the form of graphs. So current flow cytometers have the capability of simultaneously measuring multiple parameters of the cells in a cell suspension. Thus, a large number of cell specimens can be processed with a quick turnaround time. So instead of working on a single cell, we are able to do it with group of cells also. And flow cytometry is highly sensitive and can detect immunophenotype of cells in a specimen with thousands of cells. Because the technique is highly efficient, so we can detect the fluorescent fluorescent, or you can say immunophenotype within a 1,000 group of cells. So the parameters analyzed by flow cytometry include physical properties of a cell. Number one, physical properties of the cells. So you have to keep this in mind. We are able to know the size, the cytoplasmic granularity, the amount of DNA content. All these things can be counted. Even cell antigens, markers, they can also be recognized by using specific antibodies. So this is the main thing. These are the 2 points for it we are using the flow cytometry for the physical properties of the cell, as well as for identifying the markers, for recognizing the particular markers. Then by using appropriate flow cytometry can reveal the cell type. We are able to know which type of cell it is. Whether hematopoietic cell, it is lymphoid cell, it is non-hematopoietic cell. We are able to know cell lineage cell, T cell, natural killer cell, epithelial cell. Even we are able to know the age of that particular cell. Whether the cell is mature or whether it is just new one. So we are able to know the age of the cell also. So we are able to know cell type, number one. We are able to know the type of cell. We are able to know the age of cells. So flow cytometry involves the analysis of the optical and fluorescence characteristics of a single particle during their passage within a narrow, precisely defined liquid stream. So the cells will have to pass, because it's a flow cytometry. The cells will pass from the beam of light. So flow cytometry involves the analysis of the optical and fluorescence characteristics of a single particle, that may be a cell, during their passage within a narrow, precisely defined liquid stream. So cell will have to pass through the stream, basically they will have to pass through it, and at that time we will able to analyze the cell. So this is for cell analysis. This is a basic components. We have flow system, optic system, electronic system, and the computer system. These are 4 steps of flow cytometry. So this is a diagram of flow cytometry. Is it visible to all of you? So the diagram is visible to all of you. If you are not getting it today, no issues. I will discuss it tomorrow also. But if you are getting it, then it's good. Now I am just focusing on the basic diagram of flow cytometry, the whole procedure which will occur inside the machine. So there is no anything which will be there outside. We will get the results only in the diagrammatic form in the computer only. Okay. So this is the diagram on which we have to focus on. This is the only the diagrammatic representation of the inner inside of the machine. So if you are able to see here, this is a cell sample. So we have to add the cell from here in a liquid stream. They will pass from this nozzle, and only single cell is allowed to pass. Only single cell. If you are able to see, the size, the shape of the cell is also different. Like this is round in shape. This cell is little bit oval and large in shape. This cell is small in shape. So only single cell is allowed to pass through this nozzle, and when the laser is there on this particular cell, then we will get the observation. Let me revise it again. The cells are there, the shape of the size of the cell is different, but only single cell is allowed to pass, and when the beam of light will put on this cell, what will happen? We will get some observation. So there are present some, you can say, optical detectors. They will detect it, and they will put the whole result in this computer in the form of graphs only, in the forms of graph only. So this is basically the whole thing which is in the flow cytometer. So this is the inside of the machine which is there, but the main thing is this only. So there are some mirrors are also present which will enhance it. There are detectors present, filters present. All these things we will get the result only in the computer. So uh, uh, some things are there which we have to consider. There are two types of scattering which is there in flow cytometer. So physical properties such as size, like this is the cell, the physical properties like size can be represented by forward scatter as well as angle scatter. Now what will happen? We have two types of scattering in case of flow cytometry. We have forward scattering as well as we have right angle scattering. So when the beam, the light will put on this cell, what will happen? Some rays will pass as such, which is known as forward. These forward rays will be detected by forward scatter, and some rays will touch on the cell and they will move on the sides. These rays will be detected by the side scatter detector. So we have two types of detector. Physical properties like size of the cell can be detected by forward and side scatters, and internal complexity can be determined by the right angle. I will show you more. So we have two types of scattering. We have forward scattering, we have side scattering. Forward when they will pass from the cell, and side scattering when they just strike with the cell and the light will change its direction. So forward scattering. In case of forward scattering, it collects light at 180 degree from the point at which the laser beams intersects the cells, usually on a linear scale. It is correlated with the cell size and thus distinguish the cells. So this forward scattering, this is helpful in measuring the size of the cell, because only two rays are here which pass from the cell. So from this forward scattering we are able to measure the size of the cell. Either these are small cells, large cells, we will get it by the forward scattering. And the side scattering which is there, side scattering collects right angle light at 90 and it is correlated with the cytoplasmic granulity and nuclear configuration. So this side scattering, because they strike with the cell, so they will tell us, these side scattering will let us know about the internal complexity of the cell. So we have discussed two terms, forward scattering as well as side scattering. Forward scattering will tell us the size of the cell, number one. Side scattering, this side scattering, because it strikes with the cell, it will tell us about the internal complexity of the cell. So side scattering, it is it tells us the cytoplasmic granulity or internal complexity. So the combination of both forward and side scattering can distinguish normal lymphocytes, granulocytes and monocytes. Like we are able to distinguish the blood cells, the types of cells which are present in the blood. So the detection of lymphocytes and monocytes provides a reliable internal control to evaluate the size of the cells of interest. Now this is again the same thing. The beam of light, it just pass from this cell and we will get forward scattering. If you are able to understand, we will get forward scattering as well as we will get the side scattering. So if the size of the cell is large, the forward scattering is more. If the size of the cell is small, the forward scattering is small. So by this we are able to know the size of the cell. And similarly side scattering is there, which will tell us the internal granulity of the cell. If you are able to see this graph, there are lymphocytes, monocytes, neutrophils. Now the size of lymphocyte is small, so they will, in the forward scattering, they will come here. Then monocytes size is little bit more, so in the graph they will present here. And the neutrophils are quite big in size, so they will be present in this area with the blue color. So by this graph we will able to know that this cell is quite large, this is neutrophil. This cell is small, this is lymphocyte. Now the second thing is Granularity. So if you are able to see side scattering also, this neutrophil is highly granular. This is internally more complex. So it is on the higher side of the graph, and lymphocytes are present on the lower side of the graph. So we have forward scattering as well as we have the side scattering. Forward scattering tells us the size of the cell, and side scattering tells us the granularity which is there in the cell. Getting my point? Are we able to get it till now? Are we able to understand forward scattering and side scattering right now? Okay. So shall we continue now? Okay. Okay. Thank you, Vishwa. Thank you, Sandeep. So let me let me tell you again that we have two terms. We have forward scattering, we have side scattering. Forward scattering will let us know about the size of the cell. Side scattering will let us know about the internal complexity of the cell. So if you see this graph, then you will in the graph you will get only dots only. So by these dots you will able to know the size of the cell. Suppose these dots are here, that means this cell is quite small in size, only 50, and this red one is large in size. Similarly side scattering is there. So the more the complex the cell is, the graph they will present here. The cell, if the complexity is more, then the cell will be present here. If the complexity is less, then it will be present here. So this is the main thing which is there in case of forward scattering and side scattering. So from the forward scattering we are able to know the size of the cell. From the side scattering we are able to know the complexity of the cell. So flow cytometry is composed of three main components: the flow system. This is only the internal part of the machine, but I will let you know the basics of it. So flow cytometry is composed of three main components: the flow system, fluidics. So cells in a suspension are brought in a single file path. The optical system, also known as light sensing. In the flow system, the cells are allowed to pass. Getting my point? Cells are allowed to pass in a suspension, but only single cell is allowed to pass. Optical system is basically what? When we have to put the laser on the cells. So, optical system is a focused laser which scatter light and emit fluorescence that is filtered and collect. So, optical system is when the laser beam is allowed to strike with the cell. And the third is the electronic system, which is known as signal processing. Emitted light is converted to distinct values that are stored in a file for analysis. So, this is the electronic system. So one of the fundamental of flow cytometry is the ability to measure the properties of individual cells or particles, which is managed by a fluidic system. So when a sample is injected in a flow cytometer, it is ordered into a stream of single particles. The fluidic system consists of flow cells. So this is the thing. This is the fluidic system. So there are many cells, but only single cell is allowed to pass. This is also known as hydrodynamic focusing. So once the cell is injected into a stream of sheet fluid within the flow chamber, they are forced into the center of a stream, forming a single file by the principle of hydrodynamic focusing. So only one cell is allowed to pass. Please keep this thing in mind that only single cell is allowed to pass from this laser beam. Then the number 2 is optics. That means the laser beam, the light. So light that is scattered in the forward direction is known as forward scattering, and that this forward scattering, the intensity of this signal has been attributed to cell size. So this forward scattering will let us know about the size of the cell. Obviously, if the scattering is more, suppose the scattering is that much, that means the size of the cell is quite big. So forward scattering will let us know about the size of the cell, and the side scattering, it will let us know about the organelles which are there in the cell, about the complexity, the granularity of that particular cell. So this is the laser beam which will put on a single cell. After it, we will have to detect it in the detector with the help of these lenses only. So this is FCS and SCS. So this is forward scattering, this is side scattering, and this is your homework also for tomorrow. You have to tell me the basic principle of flow cytometry. You have to tell me about forward scattering as well as side scattering. So in the forward scattering, you will able to know the size of the cells. So here the size of the cell is small. These are basically RBC cells which are very small in size. Now it is on the 50 number, and some cells are there which are present on 200 to 250. These are quite large cells. These are granulocyte cells basically. Similarly, according to complexity also, some cells are very less complex. They will be in the lower side of the graph. Some cells are highly complex. They will be on the upper side of the graph. So you have to draw this graph also, and you have to let me know what is the basic principle of flow cytometry along with forward scattering and side scattering. So study of forward scattering and side scattering allow us to know the differentiation of different types of cells. So then we will able to differentiate the cell according to their size and according to their complexity. So in the flow cytometer, particles are carried to the laser, intercept into a fluid stream. Any suspended particle or cell from any cell particle or cell from 0.2 to 150 micrometer in size is suitable for analysis. So we will able to take that much size of the cells. The portion of the fluid stream where particles are located is known as sample core. When the particles pass through the laser, they scatter laser light. The scattered and the fluorescent light is collected by the lenses. Then after it, it will be detected by the detectors which are there, and then at the end, these detectors, they produce some signal which is proportional to the optical signal striking them. These are some of the applications of flow cytometry. It is highly useful in immunophenotyping, cell sorting, for knowing the DNA content in the cell, for cell cycle analysis, for apoptosis, for cell proliferation. These are some of the basics of the basic applications of flow cytometry. So flow cytometry is also known as fluorescence activated cell sorter. Now we have just first we discussed basics of flow cytometry. Now when we are using the cells which are fluorescently active, so considering a group of lymphocytes from a mouse that have been stained with the green fluorescent antibodies. So what we are using now, instead of just doing it simply, we are just using the cells which are fluorescent. Okay. So consider a group of lymphocytes from a mouse that have been stained with a green fluorescent antibody specific for CD4 and red fluorescent antibodies specific for CD8. So we are using green fluorescent and we are using red fluorescent. Both the labeled cells, now one is labeled with green color or you can say fluorescent, and another is labeled with red fluorescent. Now both the labeled cells, we have what we have to do, both the labeled cells generate side scattering as well as forward scattering when they pass through the laser beam, creating voltage pulses that are recorded by the computer. However, each labeled cell will also emit light of specific wavelength as a result of fluorescent label. For instance, CD4 cells will emit green color on the wavelength of 525, while CD8 cells will emit orange light at the wavelength of 560. So these fluorescent signals pass through the photomultiple tubes and generate the voltage pulses. So instead of using only cell, we are using the fluorescent cell. So the software is there. The software integrates all the information for a particular cell, align the characterization of the individual cells. So again, this is the same operators. We have forward scattering, we have side scattering. So the detection will be there, and at a particular wavelength we will able to get the detection, and the color will be like, let me show you the graph. We will get the fluorescent colors. So what are basically fluorochromes? Fluorochromes are the substances that can be excited by certain light source and emit a color at a single wavelength. At a particular wavelength they will emit a color. So fluorescent dyes can directly bind to certain cellular contents such as DNA and RNA and allow us to perform quantitative analysis. Now what these dyes will do? These dyes will bind with the DNA and RNA and they will give some color. So in most cases fluorochromes are conjugated with antibodies which specifically target cellular antigens or markers. So these are some fluorochromes like fluorosin isothiocyanate, phycoerythrin, PE texazeth, allophyocyanin. They are working at a particular wavelength. So they are using we are using it we are using it because some of them can be excited by same light source. The different fluorochromes may emit fluorescent signals with different wavelengths and colors. So we are using the fluorochromes also. So suppose this is killer cell, it will give the purple color, and this is T cell, it will give the red color, helper T cell. So we are just going to pass it through the flow cytometer. Laser beam will strike it, and under the computer we will get the graph. So red color is here, that means they are the T cell. Green color is there, and this is purple color, that means it is the killer T cell. So the size of the killer T cell is little bit more than the helper T cell. Getting my point? Let me check it. Are you getting it or not? CD4, CD8, these are the cells only. You have to focus on flow cytometry right now. These are the cells which are helpful, which are the part of immunity. Leave those things, just focus on the flow cytometry, the basics concept of flow cytometry. Please be focused on that only. The basic concept of flow cytometry, you have to focus on that only. Leave what is T cell or killer T cell. The main point is that we are using fluorochromes. And when we are using fluorochromes, we are getting the color. Otherwise, if there will be no color, the detection will be very difficult. So when these cells, they will allow to pass through the flow cytometer, laser beam will strike. Under the help of detectors and everything, we will get a data. So if you are able to see here, this is forward scattering, this is side scattering. So the red one are here, that means the size of these red one is small, and the purple one is here, that means their size is little bit more. And even side scattering is more in case of this purple one, that means the purple one, which is basically killer T cell, it is more complex. This is only the observation which we will get from this flow cytometer. Similarly, this is the activity of CD4 and CD3 cells also. So by these colors we will able to understand the properties of a cell basically. So this is how we have to process the specimen for flow cytometry. So theoretically, any specimens from which a single cell suspension can be generated are suitable. So you can take any cells, any specimen for flow cytometry. However, a lack of distinct antigen or markers in the cells of interest or tissue limits the diagnostic value of flow cytometry. Basically, flow cytometry is generally used for the blood cells like monocytes, basophils, eosinophils, neutrophils, as well as for the immunity cells like CD4 cells, lymphoid cells which are there, helper cells, killer cells. So basically which have a specific antigen antibody reaction, those cells are particularly used. So common specimens suitable for flow cytometry analysis include we will use blood we can use blood also, the cells which are there in the blood, bone marrow cells, body fluid cells, brain cells, urine, lymph node cells, any fine needle experience, you can take any cells. Okay, so specimen storage, but there is some way of using these specimens. For blood and bone marrow specimens, anticoagulants such as EDTA, heparin or acetyl dioxide are needed. Fresh tissue specimens are best transported and stored in sterile tissue culture medium. Although specimens may be stored at room temperature, refrigeration is preferred. So it is not that you have to store the specimens also before running this flow cytometry. So for flow cytometry analysis, single cell suspension of a fresh tissues can be achieved by mechanical dissociation. Then we have to prepare the cell also to run it in a flow cytometer. Single cell suspensions are required for optimal staining of samples for flow cytometry. The narrow bores of the sample injection needle and tubing on a flow cytometer will be easily clogged by aggregated cells and debris. Preparation of single cell suspension from solid tissue requires mechanical dissociation and or enzymatic digestion for optimal recovery of cells from the tissue. This is how the processing of solid tissue, how from the tissue we will get the cell, you will go it by your own self. This is applications of flow cytometry. So for knowing the DNA content, to measure the cellular DNA content, we are using the flow cytometry. So for that purpose, when you want to know the DNA content in the cell, so you have to use the dye which is propydium iodide that intercalate into the DNA helical system, and when you add this fluorochrome, when you add this cell in the cell sorter, so you will able to know the content, the amount of DNA in a cell. The fluorescent signal is directly proportional to the amount of a DNA in the nucleus and can identify the gains or losses in the DNA, basically the content of DNA in the cells. Abnormal DNA content, also known as DNA content aneuploidy, can also be determined. So these are some properties of this flow cytometer, that we will able to know the DNA content in a cell. So this is the S phase. During S phase, you all know that during the S phase the amount of DNA is doubled. So by running it on this flow cytometer, you will able to know the content of DNA in a cell. So this is again erythrocyte analysis, that means RBC cells. So detection and quantification of fetal red cells in the maternal blood. The use of flow cytometry for the detection of fetal cell is much more reproducible and sensitive than another test. Then it is also useful for detecting any like diagnosis of PNH, for detection of any diseases which are there. So these are the normal red cells and these are the JPA deficient cells. So main thing is that the only thing is that you have to add the fluorochrome and then you have to run it under the flow cytometry, and with the help of these graphs you will able to know the everything which is related with the cell. Okay, every problem, every disease, everything which is related with the cell, you will be able to know it. This is forward light scatter and we have side light scatter, and they will like forward side scatter, it is correlated with the cell size, so we will able to know the size of the cell. The side scatter will let us know about the cytoplasmic granularity and internal complexity in the cell. So if you are able to see, this is again this is small in size, so it is present in the lower side, and this is the larger one, which is granulocyte. It is size is also large. Moreover, it is more complex also. So you have to know about this only. So you can work on any different types of cells which are there in the body. You can work on this. But the one thing which you have to keep in mind, that we will get the result only in the form of graph. we will get result only in the form of graph. So this is flow cytometry, and this is a comparison with another histochemistry technique. But the main thing is that flow cytometry is a powerful technique for correlating multiple characteristics of a single cell. When you want to work on a single cell, for that particular purpose we are using the flow cytometry. This qualitative and quantitative technique has made transition from a research tool to standard clinic testing. Smaller, less expensive smaller, less expensive instruments and an increasing number of clinical useful antibodies are creating more opportunities for routine clinical laboratories. Basically, it is useful in the diagnosis and the management of the diseases. At last but not the least, keeping pace with scientific and clinical advancement is the need of an hour. So that is the all things which is there in case of flow cytometry. Now my question is to all of you is that we have just concerned cells here. We are just working on the cell on the single cell here. Similarly, we are working in the cell culture also. In the cell culture techniques also we are working on a specific cells, right? In the cell culture technique also we are working on the group of cells. The very first day we have discussed cell culture techniques. Now my question is, what is the difference between flow cytometry and the cell culture techniques? In the flow cytometry also we are working on the cell. In the cell culture techniques also we are working on the group of cells. Now what is the difference between both of these? This is your second homework. And what is the first question? I forget. I hope any one of you, please, you must know what is the first question. So this is the thing which you have to this is a very basic concept. I know all of you have many questions regarding this flow cytometry. I know it, but let me tell you, I just give you the basics of flow cytometry. This is my duty only. Otherwise, when you will work in the lab in these techniques, then you will able to understand it very, practically, then you will be able to understand it. And like some questions are there, how it is helpful to study cancer, monocytes. I will suggest you to just go through the flow cytometry virtually. Also you can go with on the YouTube also you can go with the virtually flow cytometry. The everything is there in my previous batches also. I also show them the YouTube videos of flow cytometry and everything. You can go with it and by your own self, and keep exploring. You are all are here. Keep exploring. Okay. So yes, the questions which we have flow cytometry, forward scattering, side scattering. You have to make the graph of that forward and the side scattering. Okay, very good. And the last question is that we have two techniques. We have flow cytometry as well as we have the cell culture techniques in which we are working on the cells basically. So what are the similarities and what are the dissimilarities in both these two techniques? In cell culture techniques as well as in the flow cytometry. So this is your homework for today, and thank you for your patience for listening to me. Even in the starting we have some buffering also, but thank you all for your patience, and thank you so much. I know some of you are not some of you, but many of you are new to it. But you have to go along with the basics only. That is my request to all of you. That is my suggestion to all of you, because these techniques are very very vast. Okay, within 1 hour, within 30 or 40 or 50 minutes, I am not able to let you know about everything regarding that technique, but I will only let you know about the basics of that particular technique, about the basics. So I know you are getting me, and I want to share everything. As much as knowledge I have, I want to share everything with all of you, but try to focus on the basics, so that when you will get the opportunity, I know many of you will get the opportunity in your future classes when you will do the PhD. At that time, if your basics are strong, then you can do anything. You can do anything if your basics are strong. So that is my suggestion to all of you. Even for flow cytometry, there is a workshop which are there continuously for 1 day or 2 day. I will suggest to you to go with those workshops also. The workshop is specifically on the flow cytometry. They will tell us everything, how to run a sample, how to detect it in a computer, how to read those graphs which are there. So I will suggest you to join any workshop. The workshop is maybe of 1 or 2 days. If you have the interest in flow cytometry, then you must go with it also. So that's all from my side, and thank you all for your patience. We will meet tomorrow with two topics more. The one is microscopy, and another is bioinformatics. After it, I think our or everything gets concluded, and then this lecture series will be over. If you have anything else to say, you are free to say, please. And thank you all for your good response in doing the assignments. Thank you all. I know you have your own busy schedule, you have your own exams, but till you are doing the assignments on time, I am feeling very grateful to all of you for your these efforts. Thank you all. The number one question is that you have to tell me about flow cytometry. Basically, what is flow cytometry? What is forward scattering? What is side scattering? And how to draw the graph? How to draw the cells in a graph? So this is all thing which is there in the flow cytometry. So this is the first question basically. Yes, I have to discuss something about Scoopus UGC care list. Yes, that is a totally different thing. But I will share again that I suggest you yesterday. I suggest you that you have to search for those journals which should be in the UGC care list or in the SCA list, because the journals which are there in that particular list, they are the authentic journals. Otherwise, there are so much fake journals. They will just ask you to pay 2000 or 4000, and they will publish your paper in 1 day or even in 2 days. But I thought this is your hard work. All of you are working very hard. So that is why I'm not wanting that you work so hard and you select the wrong journal. There are so many fake journals, because I am in touch with my previous students also. They also tell me, ma'am, some journals are there which is the copy of the original journal. So that is why I suggest you to search those journals related to your topic, which should be in UGC Care List 2020 or in the SCI list. So that is the only thing which I shared yesterday. No worries, no problem. I will share everything with all of you. But let me check your abstract today and tomorrow also. Then you have to continue your review paper, and then I will suggest you. So you have to select only 4 or 5 journals. 3 to 4 journals are okay, and from that 3 journals or 4 journals, I will tell you in which journal we have to publish our paper. Okay, so I will go through those journals again and I will let you know. Okay, so take care of yourself, and good night from my side. Enjoy your weekend. Maybe after you have your exams also. So work hard. And also I request you all that please like my videos and share my channel. Not share, even subscribe my channel so that you will get the notification when I just get live, because I am just doing two lecture series more also. So I think some things will be helpful to all of you, and I definitely need the support from all of you, and I know you all will cooperate. So that's all from my side. Thank you all for being here, and take care of yourself. Good night. To try to do the assignment on time, including you tomorrow. For tomorrow, I think we will do the timing within the noon, so that you all will be free by evening and you can do anything in the evening also. So I will try to send you the timing in the morning, so that you all will be ready along with it. Okay. So that's all from my side. Bye bye. Good night.